Thank you, Mrs. Primrose. So in this section, we shall see how colloidal probe atomic force microscopy is used to probe the interfacial adhesion in flax PLA biocomposite material. So these recently, the importance of green materials is becoming very, important to not only to the people, but also several governments are starting this green eco-friendly material initiative. Mainly in the backdrop of a large amount of pollution caused by non-degradable plastics around the world. And ocean, here is an ocean pollution from plastic trash. So, in this backdrop, eco-composite or biocomposite materials that are environmentally degradable for structural application is an area that is developing, and here is a cross-section SPM image of PLA flax biocomposite. And what you see is around is the flax fiber embedded in a matrix polymer, polylactic acid. And both these components are derived from renewable resources. They have a lower environmental impact, and finally they are biodegradable after their use. And also they pose several advantages, such as low density, because if you take a steel wire, the flax fiber have a low density, high specific strength, they are low cost and biodegradable. And in Europe, several automobile manufacturers have already started using these biocomposite for several components in their vehicles, and also they have potential marine applications. So this flax fiber is derived from this flax plant, and flax plant is also called linen, which is mostly used in our tissue. And this is a cross-section of this flax plant, and these fibers appear as bundles close to the stem. And here is a bunch of here's a single AFM, SEM image of a single flax fiber, which is around 20 micrometer in diameter. And if you see the structure of this flax fiber, this itself is a composite material, comprising mainly a primary cell wall and a secondary cell wall. The primary cell wall consists of pectins and hemicellulose polysaccharides, and it's a thin layer, while the secondary layer consists of highly oriented cellulose microfibers. And these cellulose microfibers are highly crystalline, and they are embedded in what is called encrusting polymers composed of heavy cellulose and pectins. So that is the load-bearing component of the fiber, which gives them this high specific strength, etc. For example, here is an image of this primary cell wall, a raw flax fiber, and once we do some surface treatments, we can remove, for example, with pectinase enzyme, we can remove this primary cell wall, and what we see is these highly oriented microfibrils of cellulose that are like 20 to 30 nanometer in diameter. And here is a 3D image of the fiber that shows they are oriented along the fiber axis. So if you take this flax fiber and put it in a polymer matrix, the composite properties depends on the intrinsic mechanical properties of the fiber, as well as the interfacial adhesion between the fiber and the matrix. Now, the intrinsic mechanical property of the fiber is governed by the interactions between different polysaccharides, between cellulose and pectins in the cell wall. And if you look at the interface between the fiber and the matrix, it's not a simple materials. There are several interactions possibilities between PLA and the cellulose, pectins, and hemicellulose. So how we can probe these different interactions and how we can optimize the interface and to enhance interface adhesion, finally determine the best mechanical properties for these eco-friendly materials. traditional methods are difficult because they are assembled techniques. So we need more local techniques to probe specifically interactions between these pairs. And our objective is to study interactions between model surfaces using local techniques based on AFM, which allows us to give a direct comparison between model and real systems. So here is a colloidal probe technique. So basically it involves sticking a colloidal particle of interest, which can be a polymer, microsphere, at the end of an AFM tip. And then we have a smooth film, essentially molecularly smooth films with the minimum roughness possible. And then we do the force distance measurements. So our major objective to choose this method, especially for these natural fibers, was there are controversial results in the literature concerning the work of adhesion measurements in composites, because in one lab they use one type of fiber, in another lab they have other type of fiber. The values are very scattered. So here our objective is to use this colloidal probe technique to measure the work of adhesion between cellulose and PLA, which are the main load-bearing polymers in the biocomposite material. So the CFM, the colloid probe technique, allows to selection of probes and surfaces of choice to study complex systems, and the force of adhesion can be modeled using contact mechanical theory like JKR, Johnson-Kendall-Roberts, or Dimitri-Muller-Topolov theory, depending on the case. And as I told before, the experimental prerequisite is smooth polymer surface and colloids, and calibration of the cantilevers after we stick the beads. So here is in preparation of the microbeads. In this case, we start with cellulose beds. So cellulose is a very difficult polymer for processing. They're very highly crystalline. So we start with cellulose triacetate beads. So we dissolve the cellulose triacetate, the solvent, and then we disperse them in polyvinyl alcohol with water with 2% solution with high stirring. And once we introduce the polymer solution, what happens is an emulsion solvent evaporation process, which results in giving this cellulose microbead material. We take this cellulose, this is cellulose triacetate microbeads. We take them and we do this saponification reaction by treating with an alkali solution at 90 degrees, and then finally we regenerate back the cellulose, giving these cellulose beads. And here is a infrared spectroscopy curves that shows cellulose triacetate with these acetate peaks, carbonyl peaks, pure cellulose, the pink one, and the regenerated cellulose closely matched to what is the pure cellulose, indicating that reaction was good. And once we have these cellulose microbeads, we stick to the tip of the cantilever like that to get this colloidal AFM probe. And once we have that, we can measure the diameter or the radius to normalize the adhesion force for modeling. And then the task is to prepare smooth polymer films, here PLA thin films with roughness essentially below 1 nanometer, 2 angstroms. And once we have this sphere plane geometry, we can do the adhesion force. The raw adhesion forces are measured, normalized with the radius of the sphere, and then plotted in as frequency distribution curves. Not only that, and in the morning, Dr. Cho was telling about the essential of reproducibility and repeatability of measurements, and that's exactly the reason why we tested several measurements on several different samples, and here we overlapped them to show the good reproducibility of these measurements. Now the adhesion force that we measure is dependent on the relative humidity of the environment. And here we show that when these measurements were performed at low humidity, the adhesion forces were low, indicating that the condensed water molecule on the atmosphere is contributing to the adhesion. Here is a normalized force versus the relative humidity, and we can see that at low humidity, adhesion is low, and with increasing in humidity, it is increasing, and it drops a little because at very high humidity, where you have a very high lot of capillary forces the capillary force decrease actually, because you are essentially under water layer. So the force that we measure under ambient condition is a sum of van der Waals plus capillary forces. At low relative humidity, the adhesion force is purely from van der Waals component. And once we have this van der Waals component of adhesion, and a useful parameter that we could derive is what is called the Hammaker constant, which indicates the strength of van der Waals interactions between two surfaces across a media. And for sphere plane geometry, this is a relation. And also, we could get from the adhesion force, we could use this Johnson-Kendall-Roberts theory to measure the work of adhesion. Having this experimental data, we could compare the adhesion, the work of adhesion between cellulose and PLA, and we compared with cellulose and cellulose, and clearly it shows that cellulose PLA forms a weak interface in the biocomposite. And any processing parameter should be aimed at increasing this adhesion. The practical significance of the results is here. Actually, this is the Nardan-Schul relation that relates the work of adhesion to the interfacial shear stress between the matrix and the fiber. For materials like carbon fiber, epoxy, and glass polyester systems, we have a linear relation between the work of adhesion and interfacial shear stress between the fiber and the matrix. However, that relation is not exactly true. When we take work of adhesion from contact angle measurements, for example, contact angle of PLA over this fiber, because this is a bulk technique, we have this deviation from this linear relation. However, when we use this work of adhesion from colloidal probe technique, which is more local technique, that gives a better agreement with the Nardan-Schul relation. So here we show the relevance of these local techniques to characterize interactions in complex systems. So as a conclusion, Colloidal Probe AFM shows that the weakest interface in a biocomposite is between PLA and cellulose. The adhesion forces is measured, it can be modeled using sphere plane contact, and this is not limited to this system but to any other systems which has been discussed in this floor before. For example, to study the interactions between, for bio recognition, for example. Yeah, that's what is written. Thank you very much for your kind attention. Thank you. And also I thank Dr. Eric Balnois, who is my professor and the teacher who introduced me to this AFM techniques from the University of South Britannia. Thank you.